Thank you very much. It's a distinct pleasure to be here on the stage today. SID has a really special meaning for me. My first SID was when I was first year PhD student in 2002. I definitely remember this meeting, very memorable. The quality of SID always is very high. It's always intellectually stimulating to be here to listen to colleagues' works. So having this opportunity today, I would like to talk about some of my research for which I don't have any conflicts of interest. I would like to start by reminding you that skin, which is an organ that we all admire and study, first formed as late as in Precambrian period, already in ancestral sponges, and it really had more than 580 million years to evolve into the complex and functionally diverse organ that we all know now today. And on this slide, above the section, histological section of human scalp skin, listed are the key landmark evolutionary events in vertebrate skin evolution. So the topic of today's presentation would be the crown jewel of evolution, the latest acquisition in skin, cycling hair follicles. Many of you studying hair follicles truly understand its complexity, and it would be probably correct to say what I typically say to new graduate students coming into the lab, that they should not expect to be experts in hair for at least first 3 years. So for the purposes of today's talk, I will oversimplify what hair follicle is, and I will use perhaps not very careful analogy of that of our 3D printer. The analogy of the hair follicle and 3D printer is somewhat valid, because just like 3D printer that contains the ink cartridge, hair follicle contains a very defined stem cell rich niche that supplies its multiple cycles of regeneration. And the production of the hair in form of cell-based printing takes place at the bottom of the growing hair follicle, where mesenchymal niche represented by dermal papillae, as well as proliferating matrix keratinocytes represent so-called CPU and printing unit of the follicle. So just like 3D printers, production of hair occurs in defined cycles that consist of consecutive phases. So the growth phase, when hair is being produced, called anagen, is followed by the regression phase when follicles involute and go into the resting phase, at which point they could reset the print program. And similar to printers being fed different instructions, hair follicles, same hair follicles can actually output dramatically different looking hair shafts over their lifetime. And this is really well represented on the next slide, where same hair follicles in juvenile and adult mammals in many species, such as in case of wild boar, can produce very distinct hairs, both in terms of length, texture, as well as color. And it all relies on reprogramming of the hair follicle program, which we still really poorly understand. So the production of hair follicles also does not occur in isolation. It really occurs in this large arrays of many thousands of hair follicles. And for in most animals to be what it is, the hair printing activities need to be somehow synchronized. What's important during hair production really differs between species and between body sites. So if we take purely human scalp perspective, something that's clinically very relevant, so what's really important is that anagen should be as long as possible, because what's desirable is hair strands that are very long. And telogens should be also as short as possible. And main reason for that is that human scalp hair follicles do not retain old club hairs for a very long time. So soon after entering telogen, they would shed their club hair and will be essentially empty telogen follicles. So when anagens are long and telogens are short, this is typically not a problem, as in young adults or children. But in men affected by androgenic alopecia, shortening of anagen and lengthening of telogen with the background of empty hair follicles will present itself as alopecia. But goals for hair production across the skin are actually very different in many mammalian species. So for most mammals, what really matters is that fur is very uniform and typically short in length, so that requires fairly short anagens on the order of weeks and months only. And like mice, many mammalian species actually can very effectively retain old club hairs for very long durations of time. So here I'm using northern elephant seal as a really good example of it. So this partially oceanic animal cycles only once a year during summer month. And the cycle presents itself as this catastrophic molt event. So tourists that typically observe molting seals tend to get really nervous because what it looks like at the distance is that seals are losing the sheets of skin. But what it is, as shown on the picture on the right, these are just fields of club hairs that are encased in the keratinized epidermis that come off. And as this club hair shedding occurs, new hair is being produced by the underlying hair follicle. And that hair would be sufficient to maintain the fur of an animal for the duration of the next year. And essentially, with this large-scale problem of producing high-quality fur, what's really important to understand is how long ago did this problem originate? As far as we can understand, really high-quality, very complex fur with multiple hair types already existed in now extinct mammaliforms, which are the predecessors of nowadays mammals. And here's an example of a Jurassic mammaliform that had very well-preserved skeleton and the integument imprint where it's very clear that they already had high-density fur with really beautiful, complex-looking hairs. So fur existed for a very long time. And using this slide, I also would like to take an opportunity to kind of reflect what was the life of a mammaliform with hair at the time. And this is a fantasy of National Geographic illustrators, which is fairly correct. So on a normal day, a hairy mammaliform would encounter Archaeopteryx, raptors stealing eggs, Brontosaurus, Brachiosaurus. So it was really an exciting time for skin biologists to do research because there's all this beautiful models running around. So it would be nice to go back in time. So what regulates large-scale regeneration? This question is something that occupies a lot of my thinking and my research. And what we understand is that although hair follicles intrinsically have all the necessary cellular and signaling components for to complete a cycle, typically they do not do this autonomously. Typically they are heavily influenced by signaling inputs originating from the surrounding environment, which we define jointly as signaling skin microenvironment. And what are the key components of signaling microenvironment? One of the primary components are actually neighboring hair follicles. Then there are cell types that exist outside of the hair follicle, prominently adipose, dermal adipose tissue, progenitors, and differentiated adipocytes. I will mention a little bit about the chondrocyte in the mouse ear. More recently, multiple works are emerging on the important regulatory role of adaptive and innate immune cells in hair cycle control. And the signaling inputs that hair follicles receive from their microenvironment could be both stimulating as well as inhibitory. And furthermore, signaling exchange is really bidirectional because hair follicles can likewise dramatically alter signaling input for their non-hair neighbors. And in today's talk, highlighted here in green, would be some of the aspects of signaling microenvironment that I will mention. So perhaps one of the best examples of the signaling microenvironment operating in mammalian skin is hair regeneration in rodents that presents itself as the migrating wave patterns. They can be observed non-invasively upon simple shaving of mice on the basis of skin pigmentation because conveniently in mice melanogenesis is primarily restricted to the growing hair follicles. And at the microscopic level, it really presents itself as the spatially laid out temporal wave of the hair cycle where neighboring hair follicles that enter new onogens signal to their late telogen neighbors to stimulate them into an onogen in the form of chain reaction. Which produces regenerative waves. So hair regeneration across skin in form of waves is not restricted to mice. It also exists in other species, including other rodents, rats prominently displayed, as well as non-rodent species. Here's an example of a non-rodent, a rabbit, and this is shaved rabbit that typically would be just covered by fur, but if you shave it, you will observe this very complex hair regeneration patterns. These patterns are dynamic, as one would expect, because if traced over time, as shown here at the top, the same rabbit that is now here represented in blue color, over the course of previous months actually developed this pattern from a much smaller patch at the base and near the tail. And at the bottom is another example of rabbit where hair wave patterns start in an explosive manner over the course of just 2 weeks. So what do we understand about the signaling basis of hair-to-hair communication? What's really exciting is that the earliest observations that hair can regenerate the wave really dates back to as early as 1940s. When being still a graduate student, we first wrote our review, it was trashed in a review by an anonymous reviewer to whom I'm really grateful, because he pointed out that there's this classic literature which we were not aware of, and it really made me go to a library at USC, but they actually had a collection of original nature magazines going all the way to 1800s. To 1800s, and I found the issue from 1945, and I was really amazed. This first paper by Harrow that described presence of this pattern in rats. So people knew about the existence. Mechanism was not well understood. So what do we currently understand is that for hair waves to form, the follicles engage in what's called signaling co-option. So signaling co-option is a phenomenon that essentially requires three components. First, two cell populations that interact need to necessarily rely on the same signaling pathway. Both cell populations need to reside within a signaling gradient range, which is normally on the order of 100 microns or less. And out of the two interacting populations, one should be the dominant source of the activator, so it will be the driver of the interaction, while the second population will be its recipient. So in case of the hair spreading wave, which is mediated by the canonical Wnt signaling, the driver is the early anagen hair follicle, in which Wnt ligand production dramatically increases. And the recipient is the neighboring late telogen hair follicle. The target within hair follicle for this macroenvironmental Wnt signaling is actually dermal niche, the dermal papillae, which becomes Wnt active and goes into an alert state, but yet not anagen state. To progress into anagen state, process requires additional paracrine signals that amplify the original input and convert it into proliferating hair germ, at which point hair follicle transitions into actual anagen. So the role of Wnt activation in this process could be visualized, such as in Wnt reporter mice. So shown here is the bat gall reporter, and we're looking at the very front of the hair spreading wave, and here are the micro-dissected hair follicles indicated by numbers. So you can see that the hair follicle that's still intelligent, that's closest to the wave front, number 3, has Wnt activity in dermal papillae, and just a few rows behind it is follicle entering early anagen with Wnt activities both in hair germ and dermal papilla. So for Wnt-based hair follicle regeneration to occur, what's really is critical is initiation. So initiation of spontaneous initiation of hair regeneration in otherwise telogen domain also appears to rely on Wnt signaling, but it's not very efficient. As far as we understand, in late telogen skin, multiple hair follicles stochastically enter alert Wnt active state, but they typically do not progress into anagen. So for anagen to occur, what is required is a group of at least 5 hair follicles, such as shown on the right top, where follicles go into Wnt active state, at which point the small group transforms into anagen population, and then through a much more efficient wave spreading, it will propagate anagen signal through the rest of the skin. This could be further validated, the role of Wnt signaling in this process in mutant mice, such as in the K14 Wnt7a mouse originally generated in the laboratory of Dr. Sarah Miller, where overexpression of Wnt signaling now makes mouse skin behave more like a rabbit skin, where you have many more spontaneous initiation sites appearing at once and very complex hair growth waves that collide and fuse with one another, leading to more dramatic hair regeneration patterns. So from what I told you so far, on the basis of stochastic hair cycle initiation and amplification through wave spreading, you would expect to generate fractal-like asymmetric patterns, such as an example of this shaved rabbit. But what's clear from the classic literature and from our own observation in young adult rodents, such as rat on the left and mice that we tracked on the right, during first couple hair cycles, hair growth patterns are almost precisely bilaterally symmetric, so which does not fit into the explanation of stochasticity. We employed computer model to identify what could be the parameters required to obtain bilateral symmetry, and the only condition that satisfied it had to include two interacting domains in the form of a cylinder that represents an approximation of the entire mouse skin, where one domain shown here with orange is the dorsal domain, which intrinsically cycles slower compared to ventral domain, and hair growth cycle waves originating in ventral skin will then spread over the flanks of the animal, giving an appearance of bilateral symmetry if one were to observe it from the top. And this is shown here at the bottom, where you have a color-coded simulation output, with green being the simulated onogen and red being the telogen. So in collaboration with Kirash at the University of Queensland, we employed his top flash wind reporter mouse, which provides whole skin live wind imaging using luminescence approach. And as shown here, same mouse was tracked over the period of about 48 days. And at the onset of postnatal first and second onogen, it's very clear, such as here at day 24, that onogen starts in the ventral skin. It starts in the chin and chest area. And then at day 26, you can see how it progresses over the shoulder region in form of bilateral wave. And same repeats itself in the next onogen at day 44, where ventral skin initiates onogen first and then propagates it over the flanks of the animal. So graph on the right shows the quantifications of luminescent skin imaging, with ventral skin being blue and dorsal skin being orange. And so the highlighted parts represent temporal advancement of hair cycle of ventral over dorsal skin. And this intrinsic faster hair cycling of ventral skin is actually preserved upon grafting, such as shown in this experiment, where chin as well as dorsal half-centimeter piece of skin was grafted to pigmented skid mice. So over the experimental time course, chin skin underwent 3 cycles, while dorsal skin underwent only 2 on average. So we performed bulk RNA sequencing analysis on the different skin domains from mice at various time points, and here I'm just showing what we define as refractory telogen, to which I will return just a little bit later, and this represents early telogen, and when compared on RNA sequencing, profiles of different skin domains are actually distinct. And what's really critical is that distinction are within BMP and Wnt signaling pathways at the level of multiple ligands and antagonists that could be summarized such that chin and ventral skin has more Wnt ligands, fewer BMP antagonists, and as well as other known hair cycle activators and inhibitors. Such that ventral skin appears to have more activation signals. So the key role of Wnt and BMP signaling in guiding domain-specific hair cycle is also demonstrated here using reporter mice. So at the top is the Wnt reporter mice done at the same time point between chin, ventral, and dorsal skin. And what we're looking at is that alert hair follicles with Wnt-positive dermal papillae. To kind of simplify it for you, here I highlighted them with the red, so you can see a dramatic difference between dorsal as well as ventral skin in that sense. There's many more Wnt-positive alert hair follicles in the ventral skin. While in the BMP reporter mouse, that marks BMP positive bulge stem cells, pattern is quite opposite, where there's a lot more BMP active bulge regions in telogen hair follicles of the dorsal skin. And so this role of Wnt and BMP signaling in regulating domain-specific hair cycle pace could be further validated in mutant mice when either canonical Wnt7a ligand or BMP ligand are overexpressed. So in case of Wnt7a, overexpression leads to loss of the ventral skin dominance, where spontaneous initiation sites now arise prematurely in dorsal skin, such as in the center, and upon overexpression of a hair cycle inhibitor BMP4, ventral skin dominance and bilateral pattern also gets disrupted through somewhat different mechanisms, where waves cannot efficiently propagate and they stall and thus become very fractal-like and broken. So now I would like to draw your attention to kind of like the summary model that we've conceptualized based on this. So where it's skin, you can think of a skin as a patchwork of developmentally defined heterogeneous regenerative fields where hair follicles within those individual anatomic domains have intrinsically different hair cycle dynamics. And they are primarily underlined by domain-specific differences in the Wnt and BMP signaling pathways that we are just really starting to understand. But one of the key observations is that ventral skin has intrinsically higher Wnt and lower BMP activity levels, enabling it to act as sort of de facto hair cycle pacemaker for the rest of the skin, at least for the part of the mouse's life. So what are the functional implications of this? One implication is that by having a more reliable initiation site in the skin could provide a more robust system for skin-wide hair regeneration. So you don't need to only rely on stochastic spontaneous initiation sites that I presented to you earlier. And another functional implication could be site-specific fur density. So when we measure the number of club hairs, average number of club hairs per hair follicle in 4-month-old mice, it was very clear that ventral domains, especially chin, has much higher number of so-called triplicate hair follicles where each follicle contains 3 club hairs compared to dorsal skin where the average number is 2, which means that with the exact same hair follicle density, having more club hairs will provide you a higher thickness fur, which can be beneficial for mice that are low-gated animals, and most of the heat loss actually occurs through a contact with the ground. So another really intriguing phenomenon that was already observed in 1960s and also was worked on by Ralph Poss and Vladimir Borchkarev is this phenomenon of telogen refractivity. So this is a phenomenon when telogen skin that just entered telogen, meaning right after anagen, is somehow resistant to normal anagen initiation from the surrounding. So shown here on the left is a picture of a shaped mouse where anagen is propagating from a central initiation site, but a coin-sized piece of telogen skin is completely resistant to it. What is now known as first phase of telogen that lasts approximately 30 days in mice is functionally distinct from the later phase. And it's called refractory versus the later phase that's more dispensable, and it's what we call competent. And hair regeneration induction is very difficult to achieve during this first month. So while competent telogen hair follicles could be rapidly activated to anagen, and this phase, which is now shown in dotted line, could be either really short if anagen signal is presented early, or could be actually much, much longer if anagen signal is not present in the skin environment. So follicles will be in this alert waiting state. What we understand from our earlier studies that one of the cell types in skin microenvironment that essentially enforces refractory state on telogen hair follicle are dermal mature adipocytes, which are the site of BMP2 ligand production. And by implanting, for example, recombinant BMP protein coated beads ahead of the wavefront, as shown here on the right, we can actually result in reactivation of the refractory state, and it will generate a halo of inhibition where hair follicles cannot go into anagen around the beads. Other groups are now exploring role of other macroenvironmental players such as immune cells, and clearly it's not just one cell type or not one pathway that enforces refractivity on early telogen hair follicles. So this is really a developing field. What drew our attention lately is this phenomenon of hyperrefractivity that seemed to exist in mouse ear skin. So mouse ear skin is quite unique. Hair follicles, they go through morphogenesis, they go through the first morphogenetic anagen, at which point they enter into this very long-lasting, months-long lasting telogen. They remain competent to anagen initiation because if you attempt to activate them, such as by ear-wide hair plucking or topical application of potent hair cycle activator sonic agonist, you can actually reactivate premature anagen. But things such as cyclosporin A treatment, which is very effective at inducing anagen in dorsal skin, has no effect on anagen initiation. So clearly there is something in the macroenvironment of the ear hair follicles that's causing them to become this extra resistant to regeneration under normal condition. And what's in the ear is a novel tissue type. So ear hair follicles, as shown here, coexist in very close proximity to auricular cartilage. So this is a neural crest-derived cartilage tissue, which is shown here as SOX9 positive line of cells. And what we understand from RNA sequencing profiling that we did, ear cartilage in particular is enriched for well-established antagonists of hair regeneration. So they are the primary source for the production of BMP5 ligand. They also produce a number of soluble Wnt antagonists as well as FGF18, yet another potent signaling inhibitor of hair regeneration. And just kind of a side note, BMP5 mutant mice, one of the first transgenic mice for which gene was actually defined, and these mice also called short ear mice because they have short and malformed ears. So BMP5 is a necessary autocrine factor for normal cartilage morphogenesis, and by association it also affects hair cycle dynamics in ear hair follicle. So the role of Wnt and BMP signaling could also further demonstrated in the ear in the reporter mice, such as shown here for BMP and Wnt reporter, which is either highly active or almost completely absent in ear mouse skin. And by modulating Wnt signaling in the ear, such as in K14 noggin mice, or expressing soluble antagonists for BMP noggin, or again in K14 Wnt7a mice, we can partially rescue this hyperrefractivity and allow for ear hair follicles to spontaneously cycle faster. So in the concluding part of my talk, I would like to kind of switch gears a little bit and talk about what I mentioned earlier, that the flow of signaling information between hair follicle and the macroenvironment is really a bidirectional process. It is well known to hair biologists since really early days of studying hair cycle is that what cycles almost in synchrony with hair follicles is dermal adipose tissue and dermal vasculature that dramatically expand during anagen and then reduce in size during telogen. The reason for that is, for example, in case of adipose tissue, is because both adipose tissue and hair follicle highly rely on Wnt and BMP signaling for their respective cellular activities. And this kind of symbiosis between hair follicle and adipose tissue is particularly prominent in the model for wound-induced hair follicle neogenesis. So this is an adult mouse model system where large wounds spontaneously initiate regeneration of new hair follicles, such as shown here at the top right corner. White are the newly regenerated hair follicles and mouse wounds. So this model was rediscovered and popularized by Mayumi Ito and George Katsarelis, and now is considered one of the leading models for studying adult skin regeneration. So following hair regeneration, what's really exciting is that a fairly normal-looking mature dermal adipose tissue regenerates around hair follicles. And process for adipose tissue regeneration in wounds is dependent on the hair, because over the time course, hairs form first, and for fat forms second, and for fat to form, they necessarily require hair follicles, and hairless wounds, even large hairless wounds, will not have differentiated adipose tissue present within it. At the transcriptional level, what we understand is that primarily non-competent for adipogenic differentiation, wound scar myofibroblasts acquire competence for that by reactivating embryonic transcriptional program for white adipose tissue formation. And one of the key transcription factors that they activate next to the hair follicles in wounds is this molecule called zinc finger protein 423. It's shown here in the red. This is considered one of the key commitment factors for white adipose lineage during morphogenesis. So normally it's active in the early postnatal skin, as shown here in the center panel left, because dermal adipose tissue continues to develop in the early postnatal time in mice. Then reporter activity shuts down in an adult skin, and it gets reactivated in wounds containing hairs, as shown here in the middle right. So what drives CFP is the hair follicle-derived BMP signaling, because anagen hair follicle is a prominent source for BMP2, primarily formed in outer root sheath, as well as BMP4 in dermal papillae. So shown here are the wound phenotypes of K14 noggin mice, where hairy wounds lack hair follicles because BMP signaling is suppressed. And a qualitatively similar outcome can be obtained if BMP receptor is knocked out in wound myofibroblast using SMAC3. as well as upon a systemic treatment of mice that regenerate wounds with potent BMP inhibitor LDN. And essentially, story emerges that hair follicles produce BMP-rich microenvironment in wound scars that enables adipogenic reprogramming of myofibroblasts in wound. And this process can be partially replicated in a dish when non-adipogenic early scar myofibroblasts are exposed to a pulse of recombinant BMP protein, which is sufficient to induce adipogenesis. And this is partially conserved in human system, where at least in keloid scar fibroblast, adipogenicity could be dramatically enhanced in the culture of primary keloid fibroblast, either upon exposure of cells to recombinant human BMP4 protein or to the secretome of cultured human scalp hair follicles that were microdissected and placed over the membrane. So you can also attend another talk from my laboratory by Christian that will take place on Saturday, and he will discuss some more new details about the diversity of wound fibroblasts and how different fibroblasts engage in regeneration. This will be on Saturday. And in my conclusion slide, I would like to just summarize to say that hair follicle is really the crown jewel of skin. It's the latest evolutionary acquisition, and it's tasked with a really important role to produce high density fur that's vital for the survival of most mammals. And to do so, it relies on a lot of signaling help from the surrounding hair follicles as well as the surrounding non-hair follicle cells. And this macroenvironmental regulation of hair regeneration leads to body-wide patterns of hair regeneration, and I mentioned to you site-specific differences. And with that, I would like to end and give a quick acknowledgement to former and past laboratory members. Special thanks go to very supportive computational and skin biology team at UC Irvine, whose names are here in green. My former PhD and postdoctoral mentors, Dr. Chong, Katsarelis, and only some of the closest collaborators and funding sources. Thank you for your attention.
Thank you, Dr. Pilkus. There's no discussion following the state-of-the-art lectures, but I'm sure that you will be available in the break for questions. Thank you.
Thank you.